Saremo molto stretti sui tempi, ma insomma, adesso apro il programma così vi chiamo uno alla volta. Perché è così bassa questa? È là, c'è già. C'è già. Io aspetto 2 minuti, poi cominciamo. Sì. Intanto apro. Sei tu il primo, benissimo. Ti chiamo tra un secondo, fammi aprire il programma così conosco almeno i vostri nomi. Che è successo? Perché queste luci? Ok. Sì. Sì. Adesso apro anche quello. Non ho i nomi. Sul programma non ci sono i nomi. Abbiamo, vabbè. Sì, poi chi te lo dici? Quello con i nomi, dai. Al programma?
Sì, ora guardare in fondo, c'è il monito di uno.
Ok. Eccolo lì, sì. Davide Bongiovanni, faccio l'appello. Chiara Bottaro, ok. Angela Carfora. Francesco Dattilo, ottimo. Filippo Drigo, perfetto. Non so se tutti avete caricato le slide. Ok, perfetto. Allora, possiamo cominciare. Davide Bongiovanni ci racconterà the correction between varying degrees of organization in soil antibiotic resistance. Io mi metto un timer, così quando suona sai anche tu che sono passati 10 minuti. Ci sei? Sì. Io guardo lì. Vai.
Good morning, everyone. I'm Davide Bongiovanni, and the focus of my PhD is to investigate antibiotic resistance on soil of urban park. Antibiotic resistance is a natural phenomenon. Some microorganisms evolved the ability to produce antibiotics, which are molecules able to inhibit the growth of other microorganisms. And in response, other microorganisms evolved the ability and evolved pathway able to bypass the effect of antibiotics. For example, the efflux pump, so the ability to pump the drugs out of the cell, or inactivation of antibiotics through enzymes, or target protection. So antibiotics will be no longer able to bind its target, becoming ineffective. The genes that confer resistance to antibiotics can be transmitted vertically through generations, but also horizontally through transformation, transduction, and conjugation. As I said, antibiotic resistance is a natural phenomenon, but it has been exacerbating by the overuse and misuse of antibiotics by humans. WHO estimated that by 2050, 10 million people per year could die because of antibiotics resistance. These are the reasons why antibiotic resistance is well monitored in those fields that use antibiotics: clinical setting, agriculture, fisheries, aquaculture. But it's important to approach to this problem by a One Health perspective. So what happens in other environments? We are focusing on soil of urban parks. Soil because it's the most important reservoir of antibiotic-resistant genes, and also because the microbial biodiversity is fundamental for a biogeochemical cycle. In order to achieve the 11th goal of Sustainable Development Goals, which is the creation of a better and a more resilient and sustainable cities, it's important to know the microbial biodiversity. Urban parks, because they represent an important pathway for the spread of antibiotic-resistant genes through microbial community to humans directly, or through vectors like animal pets. So our goals is to investigate both biodiversity and resistance, and also investigate if and how human activities have an impact on both biodiversity and resistance. We sample soil in 10 parks in Turin, specifically in 2 parks as a native, as a control, and the other inside the city of Turin. We investigate the environmental matrix around the sampling point through QGIS software, and we collected information on soil parameter, physical-chemical parameters, and abundance of metal. In order to obtain information on both biodiversity and resistance, we are using a complementary approach, a culturomic one and a metagenomic one. Today, I'm going to focus on the prokaryote resistance, starting from the metagenome. We use ArcZoap, which is a bioinformatician tool that, starting from metagenome, extract sequences that confer resistance to antibiotics and classify them based on mechanisms. It's important to underline that we are focusing on prokaryote resistance. So the first question is there a differentiation between native park and urban park based on mechanisms? Among different mechanisms, antibiotic efflux is the most abundant, the most present, and there is a differentiation between native park and urban park. In the native park, there is an higher abundance of genes that confer resistance through efflux, which is a generic mechanism of resistance. While in the urban park, there is a higher abundance of genes that confer resistance through inactivation and target replacement, which are a more specific mechanisms of resistance. This differentiation, of course, could be related to the microbial community. So we investigate the fungal biodiversity and the bacterial biodiversity, and we can observe that in the native park, there is a higher abundance and a higher biodiversity of fungi, while in the urban park, there is a higher abundance and a higher biodiversity of bacteria. So we have a higher abundance of genes, bacterial genes that confer resistance through a generic mechanisms of resistance. But we have a higher abundance of fungi. So going into more detail of this, we investigated which fungal genera are more present in the native park. And as you can see here, some genera are more present in the urban park, where bacteria are predominant, while Penicillium and Russula are more present in the native park. And as you probably know, Penicillium is one of the most important producers of bioactive compound. From Penicillium, we discovered penicillins, as well as Russula. So these two genera are related to the bacterial community. We performed a Spearman correlation, and as you can see here, Penicillium and Russula are correlated with some bacterial genera. For example, Bradyrhizobium, which is also producers of bioactive compounds, and also it's involved in the nitrogen cycle. So in the native park, we have an higher abundance of bacterial genes that confer resistance through a generic mechanisms of resistance, but we have an higher abundance of fungi. So we can conclude that fungi, which are an important producers of bioactive compound, exert a selective pressure on bacterial community. And only those bacteria that possess a generic mechanisms of resistance have a higher chance to survive. What happened in the urban park, where we have a higher abundance of genes that confer resistance through inactivation and target replacement, which are a more specific mechanism of resistance. And we also have a higher abundance and a higher biodiversity of bacteria. So bacteria are correlated with the antibiotic-resistant genes. As you can see here, in some cases, yes, for a lot of bacterial genera, but not for all. Rhodoplanes and Bradyrhizobium are negatively correlated with inactivation. For target replacement, the strength of correlation is less, and we can conclude this because of the intensity of the color. So, are the bacteria responsible for the enrichment of genes that confer resistance through inactivation and target replacement? In some cases, yes. For example, for Luteolea, which is positively correlated to target replacement, and it's also more abundant in the urban park. And there is a statistical significant difference between urban park and native park based on the presence of Luteolea. But not in all cases. For example, Connexibacter, which is positively correlated with inactivation, there is not a statistical difference between native park and urban park based on its presence. So there must be something else. There must be other factors, stressor or environmental characteristics that influence both bacterial community and that favor the enrichment of antibiotic-resistant genes, a specific antibiotic-resistant genes. So the research is not finished. We are investigating other parameters, for example, soil parameters. And as you can see here, Luteolea and Connexibacter are correlated with different soil parameters. And we also are considering the environmental matrix around the sampling points in order to investigate how human activities impact both biodiversity and antibiotic-resistant genes. For now, we can conclude that there is a differentiation between native park and urban park based on mechanisms of resistance, and that there are different stressors that may promote different mechanisms of resistance. In the native park, fungi promote generic mechanisms of resistance, while in the urban park, there are other stressors that promote a more specific mechanisms of resistance. An important take-home message is that urban soil is an important tank of the antibiotic resistance genes, and of course it should be monitored. Thanks for your attention.
Thank you, Davide. You were perfectly on time, so we have a couple of questions. We can take a couple of questions, if any. I have a question for you. No, I have the microphone, which is, how do you think that in urban soil, what is the selective factors that can induce more or higher antibiotic resistance?
In the urban park, there is less fungi. That's the first point. So human activities impact on the selection on fungi. And there are a lot of factors. For example, the fragmentation of the environment can exert a pressure, or an selective pressure on fungi. For example, the pollution that remain more stable in the soil, because the bioactive compounds that are produced by fungi are less stable in the environment, while the pollutants that are produced by humans are more persistent. Microplastic and nanoplastics, there are articles that are investigated that nanoparticles induced the enrichment of antibiotic-resistant genes. So there are a lot of factors.
Okay, thank you. Any more questions?
Yeah, thank you very much for the nice talk. Actually, I wanted to ask the same question as you, so I will ask another thing. I was just wondering if you found a correlation between antibiotic resistance and species richness, like the number of different bacteria. Did you investigate this?
Yes. We are working on an urban gradient specifically in order to better understand how the environment around the sampling point establish a correlation on the richness. So which are the factors that favor or not favor the presence or absence of some bacterial or fungi. So we are working on it.
Thank you. Thank you.
Thank you.
Now, the floor is to Chiara Bottaro, exploring spectrum and thermal response of forest habitats through satellite imagery. I didn't introduce myself. I'm Tommaso Paternello, a professor of zoology at the University of Padua, and chairing this session. Okay, please. Dovrebbe funzionare.
Prova? Ah, ok, grazie.
Deve essere verde.
Ora? Ok, grazie. Scusate. Good morning, everyone. I am Chiara Bottaro, and I'm going to present you a small part of my research work, which deals with the exploration of spectral and thermal response of urban forests through satellite imagery. So satellites continuously acquire images over our planet, documenting what are the dynamic processes that are characterizing it. Archives of satellite imageries constitute a meaningful pool of knowledge to help us understand how ecosystems respond and adapt to anthropic pressures, including climate change. And this understanding is key to address conservation and management questions. So throughout my presentation, I'm going to focus on terrestrial ecosystems and more specifically on urban forests. And the main objective of this work is to assess whether different forest habitats have different spectral and thermal response in terms of electromagnetic radiation captured by the satellites, and if these differences reflect ecological characteristics. To address the urban part of the forest, we decided to use the functional urban area framework, which is an harmonized framework to define urban areas all across European countries. And this framework uses population density and travel to work flows data. This framework is important because it allows international comparisons and policy analysis on topics related to urban. A functional urban area, or a FUA, as I would call it in Italian, is an economic unit consisting of densely inhabited city core and of surrounding, more extended area whose labor market is highly integrated with the city. So for our study, we decided to focus on the extended functional urban area of the city of Campobasso, the main city of the Molise region in Italy. To select the forest habitats, we use the Carta della Natura, which is a cartographic product that has been envisaged by the Framework Law on Protected Areas in 1991. And this cartography provides a legend for terrestrial habitat mapping, but its realization is demanded on single regions. So we also decided to work on the Molise region because this region has a good Carta della Natura with a good detail, namely 1 to 25,000, and was last updated in 2021, so it's quite recently. And this chart allowed us to retrieve and filter the most extensive forest habitat types in terms of area cover percentage, and after a first filtering, we could retrieve 7 forest habitat types for further analysis. In brief, even if this is in Italian, I'm sorry, the 7 forest types were the humid woods, the synanthropic woods, the Turkey oak woods, non-native conifers, non-native broadleaves, broadleaf plantations, and downy oak woods. To retrieve the spectral and thermal information, we decided to use Landsat 8 and 9 imagery, whose images have a spatial resolution of 30 meters and a revisiting time of 60 days. So we collected and pre-processed all the images from Landsat for the year 2022 through the Google Earth Engine platform. And we synthesized the spectral information into the NDVI, that is the normalized difference vegetation index, that for those who do not know, is a metric, very popular one, for quantifying vegetation health and density. And it is computed from the red and the near-infrared band, while the thermal information was directly derived from the thermal infrared band, from which is possible to compute the land surface temperature. So these are very preliminary results that kind of suggest us that in terms of mean NDVI, there are some differences between the forest types. And in specific, these differences are evident between May and November. It was not as evident for the mean temperature, but still we could see that at least 2 forest types differentiated from others, and these were the broadleaf plantations and the Turkey oak woods. So we decided to go further into detail to assess if these differences were statistically significant, and we prepared 20 random sample points for per forest type, for each forest type. So for each 2022 image, we extracted the pixel value in terms of NDVI and LST, or temperature. And what the statistics told us is that computing the 95% confidence interval on mean, that the mean NDVI for the Turkey oak is significantly higher, and for the broadleaf plantation is significantly lower compared to the other types of vegetation. And this behavior was the opposite for mean temperature. So the one that had the higher NDVI had also the lower temperature. So we can argue from this very short result that satellite imagery somehow can tell us about the difference between forest habitat types. And according to the Carta della Natura, these differences can also be explained from an ecological point of view. So for example, the Turkey oak is mainly observed on highly fertile stations and cool north-facing slopes, which also can explain the cooler temperature, while the broadleaf plantation are typically managed forest environments where human intervention is evident and undergrowth is scarce or absent. And this may cause a mixed information in the pixel. So maybe broadleaf plantation have lower NDVI because the information that is in the pixel both comes from the bare soil and the vegetation. So, however, not all habitats could be differentiated from each other, probably owing to the pixel resolution. It comes a question really naturally, and this question is, would an increase in pixel resolution, like for example using Sentinel 10-meter pixel resolution, allow greater differentiation between forest types? To answer these questions and others, the further steps that I'm willing to take are directed toward two sides. So first, to extend this analysis on multiple years, so to see if the differences that we observed for the 2022 are consistent through time, or they change, they reflect some kind of climatic trend or whatsoever. And second is to increase image spatial resolution to see if the spectral and thermal response changes changes through the multiscale. So if I have bigger pixels, what can I see in the same pixel if I'm going to finer and finer resolution? What does it change? So that's all for my presentation. Thank you all for the attention.
Thank you, Chiara. That was interesting. Questions? We have time to take one or two. What's the extent of increasing information you might expect making pixel definition higher?
I expect, like, for example, the differences between the other forest types were not significantly evident. And what I expect is that going to finer resolution, if there are differences, this finer resolution amplifies the difference that I can observe between the types.
Nevertheless, with this resolution, you did find some big differences.
Yes, but probably just because the two forest types that were most different are really different from each other and from the other types.
So a finer resolution will help to understand a finer scale. Okay. Yes. Questions?
Thank you, Chiara, for your presentation. And I was wondering if maybe also in the context of, I guess, resolution, it could be possible also to investigate the response not just of the forest, but of the urban areas maybe near to the forest. And if in that way it could be possible to entangle the information between responses of the forest and let's say the coupling with the urban areas where the soil is completely different.
You mean using like urbanization gradient, or you mean like forest close to urbanized area?
No, I mean how to say? Yes, forest near urban areas.
The main problem I am encountering in dealing with urban forest, let's say, is that urban vegetation is highly fragmented. And so a 30-meter pixel or a 10-meter pixel doesn't really allow you to look at single trees, which is usually the case in a urban setting. Specifically, as you go more into the center of the city, you have smaller and smaller patches of vegetation. And so I have already with big extension of forests, I have the problem of the mixed information in the pixel. So I have an information that is not pure, let's say. So it's a mixed one. It comes a little bit from the soil, a little bit from the vegetation, and so on. For sure is what I am willing to investigate more, but there is a big constraint about the resolution of the pixel of the images you are looking at.
Okay, thank you.
Okay, thank you, Chiara. That was nice. Now, Angela Carpora. Differential expression of genes involved in biotic and abiotic stress in Raphanus plants collected in human and extra-human urban areas. Please.
Hi, my name is Angela Carfora. I'm a PhD student at University of Naples Federico II. My project belongs to Spoke 5 that focused on urban biodiversity. And in particular, I study differential expression of genes involved in biotic and biotic stress in Raphanus plant, collected in both urban and exurban areas. The aim of my work is to assess the impact of intensive human activities on natural ecosystems and how global urban growth influences the adaptation of species living in urban settings, leading to modification in the gene expression profiles related to biotic and biotic stress. To explore these dynamics, we choose Raphanus raphanistrum as our biological model for its ability to survive in different conditions and environments like barren, saline, and arid lands. This adaptability allowed it to develop a robust set of resistant genes and making it an ideal model for studying how urban stress influences plant condition and gene expression. I collected Raphanus raphanistrum leaves from 8 different locations in the province of Naples. Categorized in urban, agricultural, and semi-natural areas based on Correlane cover classification. Correlane cover classification is based on consists in 44 classes. However, we summarize them in 3 classes. The selected areas are also different for soil type, limestone, tuff, and lava, and have also a different level of herbivory assessed by examining the leaf integrity. I collected the samples in March 2023, ensuring the samples were in the same reproductive stage. And I preserved the leaves in RNA later for subsequent RNA extraction. In total, I collected 208 samples. I extracted RNA from leaves of 5 samples for each population and of 10 samples for San Giorgio and Vesuvio population, because in this population I found different level of herbivory. And RNA extracted was massively sequenced by a company Novogene, and I set a bioinformatic pipeline to analyze the raw data obtained. The first steps of pipeline is cleaning, consisting in cleaning and quality checking the reads. Then I mapped the reads on Raphanus raphanistrum reference genome. I generate the count matrix at both isoform and gene level, and then I conduct differential expression analysis using a jar. And finally, the data were interpreted, clustering the transcript differentially expressed based on their function, visualizing them with different colors. In this slide, shows the resulting data obtained by comparing the gene differentially expressed between urban, the sample collected in urban and semi-natural areas. So on the left of the slide, we can view the transcript up regulated in urban area. For example, there is a high number of transposase that we know can participate in gene regulation by influencing the expression of NIRB genes as a stress-responsive motive. We can view also a higher expression of two members of mate family involved in detoxification process in plant. And for example, in Arabidopsis, two mate genes are involved in the elimination of toxic effect of heavy metals such as cadmium. As expected, there are also a high number of transcripts related to abiotic stress. For example, encoding LIA protein involved in stress defense of plant from damage caused by environmental stress. Or, for example, a sulfur deficiency-induced protein involved in sulfur homeostasis in sulfur-deprived condition by down-regulating glucosinolates. And in pink, I reported transcript related to biosynthesis of plant metabolites like flavonoids. On the left, per voi, okay, for you on the right, I reported transcript down regulated in urban area. For example, WT1 is involved in vacuolar auxin transport, but it's interesting that in Arabidopsis, the loss of function of this gene is involved leads to enhanced resistance of a broad range of pathogens. And there is also a member of Serpin family involved in defense of plant against pathogen, in particular in cereal crops. These dice show the preliminary result and the comparison between samples with low and high herbivory level. So, in the right, we show is represented the gene up regulated in a low herbivory level, and we can observe the presence of transcript related to biotic response against pathogen. For example, transcript regulated PGI protein involved in defense against fungal pathogen, or a member of family phloem protein 2-like. There is a family, the family of proteins associated primarily with phloem tissue, and the lectin within this family are involved in defense by binding the chitin cell wall of pathogen. On the right, the gene down regulating in low herbivory level, we can observe again a higher number of transposase and retrotransposase, and also transcript related to abiotic stress, for example, a member of Wuschel-like Homebox family, plant-specific transcription factor involved in a different process like embryogenesis, lateral organ development, and as well as abiotic stress tolerance. These are only preliminary results that I have to validate and investigate. Among the short-term next steps, I will perform gene expression analysis also in sample with collecting in different soil composition, so limestone, lava, and tuff. I will to validate my in silico analysis results through qPCR, and finally, since my RNA results are obtained from RNA from single individuals, I will apply a variant calling pipeline to identify eventual polymorphism related to adaptation to abiotic stress to environment to urban environment. Thank you for attention.
You as the others are on time, so we have the possibility to take a couple of questions. Yes?
Thank you very much for the talk. Maybe you said it, but maybe I missed it. Why are you so interested in the Raphanus specifically? What kind of research questions led?
Because Raphanus raphanistrum have ability to survive in different conditions. So he grew up in urban settings and also in natural, semi-natural and agricultural areas. So it's very interesting to study for a differential expression for investigate of adaptation of the species.
Do you have environmental parameters? I mean, the different areas differ by known value for the environmental factors. So to correlate differential expression to some environmental data?
The area are different because obviously the area of urban area have pressure different between semi-natural areas. The parameters are difficult to individuate for a single pressure.
Well, it would be important to have some, let's say, stressor, for instance, if you have an overexpression of xenobiotic genes. So to say you have a higher concentration of some pollutant, and then you have a higher expression of this group of genes. So it's better to have a direct correlation between some environmental parameter and the expression profiling. Otherwise, it's known that in that area there is a higher human pressure or urban pressure, but you have to define better to correlate and give a sense to the value, the gene value.
But we investigate how these species adapt to urban settings. So we observe the post. We observe the gene expression, and we observe how these species adapt to.
Okay. Thank you. Now we have Francesco Dattilo, Plankton Biodiversity in Mediterranean Sea, a trait-based model.
Hi everyone, thank you to be here today. I'm Francesco Dattilo from OGS, Trieste, and today I will talk about my latest work, that is a trait-based oceanographic model for the Mediterranean Sea. So, how many people here, by raising the hand, work with plankton, just to know? Okay, nice, I'm not alone. Good things. Anyway, just to give a bit of context, plankton organisms are really interesting because they present a highly expressive functional diversity, but also because they are really fundamental for the, let's say, ocean-atmosphere interaction thanks to their production of oxygen for phytoplankton and the ability of stocking CO2 in the sea. So it's really interesting to study this kind of complex community. And so I hope that at the end of the presentation, the interest in plankton will grow. Maybe some of them will choose to change your topic. Anyway, my research question is how much does the functional diversity of plankton impact the system in terms of functioning and community structure? So I want to understand how it can influence the food web structure in plankton community. And to do so, I implemented a biogeochemical model for the Mediterranean Sea. What I'm talking about. The model is basically able to represent all the biogeochemical interaction and all the ecological and biological part in the community, so phytoplankton, zooplankton, and bacteria that interact together. But also, it has to represent all the physical elements in the sea. So we have wind stress, horizontal and vertical circulation along and across the water column. We have rain, light interaction, and also all the, heat fluxes. And also my model take into account river runoff and dry atmosphere deposition, because Mediterranean Sea is really sensitive to the Sahara deposition of dust from the desert. Okay, so the model tried to take into account all of this together in order to represent as realistically as possible all of these really complex processes. But talking about the ecological part of the model, we created two different ecological setups. One of these represents the ecological community with a small number of plankton functional types, so different kind of plankton that differ depending by their traits, so nutrient uptake, size, different kind of motility, and so on. In particular, with the small community, we have only one type for bacteria, 7 for phytoplankton, 1 mixotroph, and 4 zooplankton. And the idea now is to enlarge the biodiversity resolution of the model. So passing from a small community to a huge, complex network. Indeed, we have the same plankton community, but represented, that try to represent more in the detail the functional diversity that can have this complex community. Okay, now the work is in progress, so please feel free to share every kind of feedbacks and every kind of ideas. But for now, I'm quite proud of the first results that are we are in the validation part, let's say, of the model. But as you can see here, okay, here we have looking at the dynamic of the chlorophyll during the different season, during the year, depending by the depth. Why I'm showing you this? Because some processes that is not so easy to represent by model is the so-called DCM, the deep chlorophyll maximum that you can find during summer. And I'm pretty proud that my model is able to represent this, that is an emergent feature. I never told to my model to represent this, but this is emerging. So, and you can see also the correct seasonality of phytoplankton. And just to investigate 2 different spots, I'm showing here the South Adriatic and also a different scenario in the South Ionian Sea, but with still the same trend and the same seasonality. This is a good thing because it's telling us that our model is going in the good direction. But I don't want to stop here, because the idea now is to investigate different sub-basin, different areas, and to study the dynamics of the trophic web. Here you can see the community with 50 plankton functional types that interact together in 2 different areas. One during the bloom, so in really rich nutrient areas, and another one is in oligotrophic areas. Why I'm doing this? Because I'm interested in investigate the inner mechanism that shape the community in the system. And this model could be really a useful tool for investigate these processes. Okay, so just to give a brief conclusion and just to point out that this model is the first three-dimensional biochemical model with this kind of biodiversity resolution, let's say, in the Mediterranean Sea. So this is the first occasion that we have to study this complex community in our sea. And our model can realistically describe the community. So it can really be critical to understand ecological mechanisms that shape plant community in the sea. So I have finished, and thank you for the attention.
Thank you, Francesco. Nice talk. Questions?
Okay. Good morning, everyone. Thank you very much for the presentation. It was super interesting. I would like to ask you, when you, let's say, when you increase the diversity of your community and you have that trophic web that you showed us with like maybe 50 nodes or something like that, how do you model the interaction between each couple of nodes?
So basically, the ecological model is built with the first trophic network. So every predator has different prey with different preferential prey. And this is, let's say, the starting weight that you give at your network. After that you reproduce the dynamic of this network, the model is able to track the information about carbon fluxes from one prey to the predator. So now enabled at every time step to know the carbon fluxes and then to rebuild the network in that specific time step. And this is important because usually we tend to think a trophic network from a statically point of view, and this gives us the opportunity also to study the evolution in time. And with this model also the evolution in space. So thank you for the question.
Yeah, exactly. That's that's the point. I mean, because when you have this so much interactions, I think maybe hundreds of interactions, hundreds of lines between the nodes. If you change maybe the parameter, the strength of the interaction, the outcome in terms of plankton dynamics can be radically different, I think. So, do you have the data for each, let's say, interaction? Do you know, like, each predator? Do you know the diet and so on?
Yes, in the sense that these kind of setups are taken by previous work of other people. So this the representation of the community is not just, let's say, random, but is taken from previous studies, and they have a ecological and biological mean. But also this model is quite interesting because it's really flexible. So I showed you a 50-node network, but if we have the data, we can also build like 1,000 of nodes in the same community. Of course, this is an issue. It's something that we have to take into account. But these setups are taken because they have the ecological meaning. Thank you.
One more question.
Thanks for the speech. It was really interesting. Is your model validated on some data, at least in some part of it? Like the level of chlorophyll is just an output of the model, or do you validate in some way with satellite imagery or something like that?
Thank you very much. Really good question, because why I show you in some moment the surface chlorophyll? Because it's the only element that we can use to validate, because all of these models are validated thanks to remote sensing of chlorophyll see from the satellites. And that's why they are working on this. These are some issues because when you represent a huge community, you cannot really know if, I don't know, a certain kind of diatom is producing exactly that kind of concentration on chlorophyll. So it's the unique way to validate them, but at the same time is still really a poor way with scarce resolution. There are some progress in this using optic to, because different phytoplankton can do photosynthesis with different intensity and different wavelength of light, so they are working a lot in also in optic, and in my groups, people such as Paolo Lazzari and Dave Alvarez are working a lot on this. But yes, it's a complex field work. Thank you for the question.
Okay. Thank you.
Good. Thank you, Francesco. It was a very interesting talk. So, last talk for this session is by Filippo Drigo. The title is Integrating Morphological, Physiological, and Molecular Approaches to Monitor Photosynthetic Biodiversity in the Venice Lagoon. Please.
Thank you. It's red. È rossa. Grazie. I'm from the University of Padua, and my PhD project focuses its attentions on the primary producers of the Venice Lagoon, and it's divided in two main parts. The first one is to study the species that inhabit this environment, and we are focusing on the primary producers since they are at the base of the trophic web, and we want to monitor them with both a morphological approach of identification, but also integrating these data, this morphological data, with molecular ones, so DNA barcoding, for example. And the other parts of the project is aimed at studying the gene expression profile and the physiological responses of selected species to heat stress, due to the fact that in future environmental scenarios, this environmental parameter will affect the communities. Considering the first part, we started with an analysis of the known photosynthetic biodiversity from the 19th century till the first 2 decades of this century. In this tree, you can see the representations of all the species that have been recorded in the Venice Lagoon. And in particular, the size and the color of the node represent the species found for each taxa. And here we can see till the rank of the families. So we can see that there is a lot of biodiversity, in particular in some groups, but we have a huge number of species that needs to be monitored and preserved. In the table, instead, I reported the absolute number of the species for time series. Moreover, in this graph, you can see the variation in time of the different taxa of photosynthetic organisms. In particular, charophytes shown a decrease during the time series considered, and with only one species recorded in the last 2 decades. Chlorophytes showed practically no variation. Heterocontophytes decreased, while rhodophytes increased throughout all these years. And we tried to use also tried to use this data to assess the ecological status of the Venice Lagoon in the considered time. And we used the macroalgae quality index, which categorized which assigned to the ecological group a defined score. In particular, we have a score of 2 for sensitive species, a score of 1 for indifferent species, and a score of 0 for opportunistic species. And in this graph, you can see the variation of the 3 groups throughout the years. And in particular, we see a decrease of score 2 species, so of sensitive species. We also use the ecological evaluation index, which divides the groups in ecological stage groups. The first one includes perennial and late successional species. The second one, the opportunistic species mainly. And again, we see the variation of these 2 groups In particular, we have again a decrease. We have a decrease of the first group. So both indexes indicate that there is a decrease of species with high ecological value throughout the years. All these data are part of a review that have been submitted 2 weeks ago to Frontiers in Marine Sciences. Considering the actual biodiversity, last year we monitored 51 different sites in the central and southern part of the lagoon, both on wood pools and some marshes. And this year we started a new campaign to cover also the north side of the lagoon, concentrating our efforts on less sites, but that will be followed throughout all the seasons in order to cover all the year and have a better view of the biodiversity of this environment. And all this data with the species recorded for each point are georeferenced. Considering the results of last year's samplings, we identified with the morphological approach these species for each taxa. In particular, we could find 4 species of phanerogams, which were Zostera marina, Zostera nolti, Cimodocea nodosa, and also Rupia marittima, and almost the same number of species for the other taxa of photosynthetic biodiversity. We also applied the DNA barcoding for some samples, and here I reported the marker used for the identification. We analyzed 81 different samples, and unfortunately we couldn't obtain a good sequence for every samples. Only half were good to be run on BLAST. And comparing the identification with the molecular and the morphological approach, we found that in 27% of the cases, the 2 approaches indicated the same species. In 34% of the cases, we had that the molecular had a higher taxonomic resolution, since based on morphological traits, we stopped at the level of genera, for example. While in another 27% of the cases, the 2 approaches indicate different species. This could be also due to my experience on the morphological identification or other traits, and the frequency was almost the same between algae and plants. However, we also found that a percent of the sequences haven't any reference on online databases, and these were of only algae. Moving to the second part of the project, we are focusing our attention on studying the heat stress response of Sporobolus marittimus, which is an autochthonous species of the Venice lagoon, and Sporobolus anglicus, which is an invasive one. And they are 2 halophytes that inhabit the margins of salt marshes, and with their root system they also had to stabilize this environment by preventing also retaining the sediments. We will study their response to heat wave both in the lab and also in the field. In the lab, because we wanted to have only one variable, which is the temperature, in order to understand better what are the effects of temperature on the species, and also in the field to understand how effectively they behave in a real environment. However, the first step to work with these 2 species is to correctly distinguish them, because Sporobolus anglicus derives from an hybridization of Sporobolus marittimus with an Atlantic species, which then duplicate its genome. So they are related, and with the DNA barcoding, it's difficult to distinguish them. So one way is to approach them with the cytofluorometric identification, since Sporobolus marittimus has half the content of DNA in their nuclei respect to Sporobolus anglicus, and here we can see the 2 peaks that of the leaves of these 2 species, and in particular we can see that the peak on the left has half of the fluorescence of Sporobolus of the peak on the right, and so we can distinguish them in this way. So after correctly identify them, we were able to perform, simulate the heat wave in the lab, and we started with collecting them in the field, acclimating them for 10 days to experimental conditions that simulated the environmental temperature when we collected them, so 25 degrees during the day and 23 degrees during the night. We then sample them at the T0 before increasing the temperature to 38 degrees during the day and 34 degrees during the night for 5 days, and sample them again 3 times during this time series, and then slowly decrease the temperature to initial conditions to see if there is a recovery after the stress. We will monitor the variation in pigment content, the stability of the photosynthetic membrane, the photosynthetic efficiency, the antioxidant defenses, and also the gene expression profile. And we have some preliminary results regarding the photosynthetic efficiency, which has been recorded with the multispec. In particular, we have here on the left, the white boxes are those of Sporobolus marittimus. The boxes in gray are of those of Sporobolus anglicus, and we can see that between T0 and T3 there is a significant difference for both species, while at T5 we have that Sporobolus anglicus doesn't have a significant difference for the FFU over FM parameter. And for Sporobolus marittimus we have a huge variation of this parameter, and this was caused by the fact that Sporobolus anglicus practically wasn't affected, since it was green and fine, while Sporobolus marittimus was dying at the end of the experiment with only some leaves that were green. Only, for example, there were some plants with only 1 green leaves and all the other part of the plant was dead. So I took the data from all the leaves that I could, so green and also yellowish one, and this was the cause of this variation that is represented here. The same pattern happens also for the quantum yield, which is the efficiency of the photosystem, and again we have that between T0 and T5 there is a significant difference again for the autochthonous species, but not for the invasive one, and the same also for the non-photochemical quenching, so the energy dissipated as heat, and also for the non-regulatory energy dissipation. So in conclusion, we can see that regarding only this parameter, Sporobolus anglicus can cope better with the heat stress than Sporobolus marittimus, since we can see that there is a recovery of all the parameter at T5. So next steps for the project will be to continue and finish the sampling of this campaign to monitor the variation of species throughout the seasons, and also finish the analysis on the data on the samples collected in the lab, and also follow a real heat wave in the field. Thank you.
Thank you, Filippo. Perfectly on time. Questions? Please.
Thanks for the presentation, very interesting. And I was wondering, in your opinion, why would the Anglicus, which is a species that is native for like northern latitudes, be more like to cope better with the heat waves compared to our autochthonous one?
This is why we are studying them, because we are trying to understand how it changed after the hybridization, its origins, because it has traits from Marittimus and Alterniflorus, which is a species of the United States coast, like we saw yesterday. And yesterday we saw that also Alterniflorus has problems in coping with heat stress. So probably with the DNA duplication, there were some factors that enable these species to cope better with the heat stress, but we don't have data. The only transcriptomic data of this group of species are mainly for salt stress, so not heat, and practically this. There was an experiment some years ago that studied the behavior of Marittimus and Sporobolus patens between cold heat wave, normal temperature, and heat wave, and again they saw that Marittimus can cope with the heat stress, and for Anglicus, we have a lack of data. We don't have many informations. Before this experiment, we also tried to expose them to 42 degrees for 5 days, but both died. So it has it has a threshold, but almost. But we think also that the tide has an important role, because last year we tried to to simulate an heat wave in the field with the greenhouses, but all the time, since they inhabit the margins, the tide covered them, so also this has another role. So probably in the field there are no differences, but these preliminary results can indicate that in future years, if, for example, the Mose is closed for 5 days during a heat wave, we can have the disappearance of Marittimus. For Anglicus, we cannot say for now how it responds, but hopefully with this experiment we will have some indications.
Hi. Hi. Thank you very much for the nice presentation. Just curiosity, why are you stressing the plants to 40 degrees? What are you trying to recreate with this high temperature? Because I don't expect 40 degrees in the Venice Lagoon. Maybe I'm mistaken.
Next, since future scenarios with the IPCC forecast, we will have an increase of the means and also heat waves. So heat waves are temperatures above the mean, but we don't have an indication on how much. So it can also be that we will have 42 degrees. We also decided at first this temperature because another work exposed these species to these temperatures, but they are mainly very high to also understand which is the highest threshold. We then decreased to 38 degrees, both because the trial, they were all dead, but also because looking at the last year recordings of temperature, the highest temperature recorded by the ARPAV throughout the whole lagoon was 37 degrees. So we then decided to also decrease in this way to simulate better. Probably in the future they will be higher, but we don't know.
Thank you for your presentation. And sorry, I have a question. Yeah. And you already spoiled a bit my question, but I was wondering if it's possible to also take into account the Mose effect on these time scales and these resistance for your species that you studied, and maybe if it's also possible to create an experiment which takes into account Moses effect, for instance, limiting the nutrients concentration or the gases inside the water that you use.
I'm not a modelist, so regarding the models to simulate the impact of Mose, I don't know. So these experiments can give us some little data that maybe with other works, considering also fish communities and other communities, to understand how the whole lagoon will respond. For experiments to simulate the real impact of heat waves on these species, the problem is that probably we still don't understand how Mose influenced the whole community, the circulation, because for now we are closing it some days, not very frequently, so the impact is probably not significant. In the future, we need to understand that, but for now we don't have data since it's some years that it's working, so we are still lacking on that part to understand and simulate it better.
Thank you, Filippo. That was nice. And the session is concluded, and we have a coffee break now. And after that, we have a poster session, and then we have a round table. At what time, round table?
Yes. So the coffee break is in that room, and we are not allowed to go outside because there are the visitors of the botanical garden. So please stay inside. And then the students for the first poster sessions are required to be at their poster. and enjoy the poster session, and then we come back and discuss.
12. We will meet again at 12:15.